Okay, so it's good. I keep saying Assalamu alaikum and I'm again saying Assalamu alaikum. I'm Dr. Rumina. I'm a molecular pathologist, molecular biologist from Shaukat Khanum Hospital. And with me is Dr. Tabinda. She's the consultant radiation oncology. So हम अपना session start करते हैं. With that, we will invite the first speaker, Syed Muhammad Adnan Ali from Aga Khan University, Karachi. He'll be talking about elevated HER2 expression is correlated with poor survival in oral squamous cell carcinoma patients. Before the session starts, I would request everybody to turn off the volume of their cell phones. Thank you. If he's not here, can you?
So, Bismillahirrahmanirrahim. Thank you very much, Dr. Tabinda and Dr. Rumina, and welcome everybody. So I am going to present our study, contribution of germline PALB2 variants to an unselected and prospectively registered pancreatic cancer patient cohort in Pakistan. This study was conducted at Basic Sciences Research Department of Shaukat Khanum Memorial Cancer Hospital and Research Center. So pancreas is a part of digestive system. It functions as exocrine gland as well as endocrine gland. And there are three main parts of the pancreas. These are head, body, and tail. And majority of the pancreatic cancer, these are occur in the pancreatic ducts. These are pancreatic ductal adenocarcinoma, which constitute about 90% of all histopathological types of the cancer. So this is the pancreatic cancer epidemiology. You can appreciate that the incidence and mortality rates, these are almost equal. It means the cancer, pancreatic cancer, it's very lethal cancer. And its prevalence is more common in Europe, America, and least common in Africa and the Asian countries. So it's seventh leading cause of cancer-related deaths, and 5-year survival rate is very low, that is up to 9%. In Pakistan, in year 2022, 1,116 pancreatic cancer were diagnosed, and of them, 97% were dead at the same year. So several genetic and non-genetic risk factors are involved in the etiology of this disease. Of the genetic risk factors, several genes have been identified. Of them, PALB2 is one of the crucial genes. PALB2 acts as a DNA damage repair pathway. And along with BRCA1 and BRCA2, it repairs the damaged DNA in the cell. If there is a mutation in the PALB2, then those individuals have increased lifetime risk of breast cancer up to 60%, and ovarian cancer up to 5%, as well as pancreatic cancer up to 10%. So PALB2 is located on chromosome 16. It contains 38 kb DNA, and there are 13 coding exons of this gene, and that code for 1,186 amino acids. And in the protein of PALB2, there are several functional domains. These domains interact with several other proteins and perform the function. So this is what is already available in the literature. PALB2 variants have been extensively studied in Europe, USA, and Canada. However, in Asian countries, only studies are available from China, Japan, and Korea. And among unselected pancreatic cancer patients, the PALB2 variants have been reported up to 2%, whereas the prevalence of pathogenic variants is high, up to 3.7% in high-risk familial pancreatic cancer patients, whereas no previous study is available from Pakistan. So the objective of our study is to investigate the prevalence of germline PALB2 variants in Pakistani pancreatic cancer patients, and then to analyze the potential deleterious effects of novel missense and intonic variants using in silico bioinformatics tools in comparison with the healthy control population. And then to analyze the clinical and pathological features of pancreatic cancer patients harboring PALB2 variants and to perform genotype-phenotype correlation. So this is a cross-sectional non-interventional case control study, and study included 150 unselected pancreatic cancer patients. These were enrolled at Shaukat Khanum Memorial Cancer Hospital and Bahria International Hospital, Lahore. And the selection criteria was adult pancreatic cancer patients, which were histopathologically confirmed. And 200 healthy controls were also enrolled alongside the cancer patients. And healthy controls, these were also adult having no personal or family history of any malignancy. And this study was approved from Institutional Board of Shaukat Khanum Memorial Cancer Hospital. So blood sample was collected after informed consent. Genomic DNA was extracted from all patients, and the complete coding sequence along with intron exon boundaries of the PALB2 were amplified, and mutation screening was performed using denaturing high performance liquid chromatography. And those variants which were detected by DHPLC, these were sequenced to confirm the variants. And then for novel variants, in silico analysis was performed to assess their pathogenicity. And those with potential effect, these were further screened in 200 healthy controls. So in a period of four years, 393 pancreatic cancer patients were identified. Of them, 243 were excluded. The reason being deceased before consent, and then consent refusal, and then some patients were lost to follow-up. And a blood sample was available for the eligible 150 patients, and these were all screened for PALB2. So these are the characteristics of the study participants. Most of the study participants were male and belonged to Punjabi ethnicity. 36% had family history of breast, gastrointestinal, and other cancers. Majority of the pancreatic cancer was occurred at pancreatic head, and the grade 2 and grade 3 disease, and the tumor size was predominantly of 2 to 5 centimeter. And majority of the patients have no perineal invasion, and the disease was unresectable for 56.7% of the patients. So this is the molecular analysis results. This is a DHPLC an elution profile. Along x-axis, there is a time. Along y-axis, there is the intensity of DNA in millivolt. And you can clearly differentiate the elution profile of the patient containing the variant from wild type. And when this patient sample was sequenced and we find a missense variant, this is shown over here. There is a single peak of G. There is an overlapping peaks of G into A. And this is the chromatogram in reverse strand. These are the total PALB2 variants which were identified in our study population. In total, 22 variants were identified, containing 10 missense variants, 3 silent variants, 6 intronic, 1 variant in 5' UTR, and 2 variants in downstream of the 3' region of the gene. Of the variants, 5 were novel. These are highlighted in blue, and 17 were previously reported in the literature. 6 missense variants which were novel, and intronic variants 2, and downstream variants. These were further analyzed by in silico tools to assess their pathogenicity using the WARCARD in silico prediction model. This is freely available on internet. This tool utilizes 23 different in silico tools and predict a score that is relevant for the disease association. The score 1 is most disease associated, and score 0 mean benign variant. So these 2 missense variants, these were having score of 0.61 and 0.78, and these were extreme variants, mean they have disease association. And using the ACMG AMP criteria, these variants were classified as likely pathogenic. And these 2 non-coding variants, which are at the downstream of the gene, 3' region, these were also classified as VUS, mean they have they may be associated with the disease with some benign scores. So these are the characteristics of the PALB2 variant carriers. The likely pathogenic missense variants, these were identified in male patients. Each were diagnosed pancreatic cancer at 48. Both belong to Punjabi ethnicity and having a family history of breast cancer, and the other patient, this one, have family history of esophageal cancer. And this VUS, this is downstream variant. This was detected in a 41-year male patient having no family history. And the other downstream variant was identified in 34-year-old female patient of Pathan ethnicity having a family history of lung in 2 individuals. it's important to show here that all these variants were detected in those pancreatic cancer patients having ampullary adenocarcinoma. So in summary, this is the first study to analyze the contribution of germline PALB2 variants to Pakistani pancreatic cancer patients. And likely pathogenic variants were identified in 1.3%, and variant of unknown significance also identified in 1.3%. No equivocal pathogenic variant was identified in our study cohort. And likely pathogenic variant of unknown significance carriers, they have diagnosed with pancreatic cancer significantly at younger age. And all likely pathogenic or variant of unknown significant carrier had tumor in ampullary region. And this is also the first report in the literature we have published that the PALB2 variants are associated with ampullary carcinoma. So we are thankful to the study participants. This study was conducted under supervision of Dr. Muhammad Usman Rashid. And we are also thankful to the clinicians and the study team. And we published this study in a well-reputed journal, Hepatopancreatobiliary Journal, and with impact factor of 3.84. And this is also the part of this study, also part of my PhD thesis. Thank you. Any question? जी. Okay, the gold standard for identification of any variant in a gene is DNA sequencing. That is recently replaced by NGS. But NGS variants should also be confirmed by Sanger sequencing. But we used DHPLC as initial screening methodology. DHPLC have up to 97% sensitivity to detect variants. But in our study, for every amplicon, we used positive control. That means we were 100% sure that every amplicon was completely resolved. So we can say that in our study, DHPLC was sensitive to 100%. So any question? Okay. Sure. Thank you.
Zia. We will now invite Minahal Zia, National University of Sciences and Technology, Islamabad, Pakistan. She'll be talking about unraveling ESR1 mutations in breast cancer patients from Pakistan, implications for therapeutic advancements.
Assalamu alaikum, everyone, and greetings to everyone attending this session today. My name is Manahal Zia, and I'm a bioscience graduate from National University of Sciences and Technology. And today I will be giving a brief presentation on my research titled Unraveling ESR1 Mutations in Breast Cancer Patients from Pakistan, Implication for Therapeutic Advancements. These are all the contents that I will be discussing today. So starting off with a brief introduction, according to the International Agency of Research on Cancer, Pakistan reported an astonishing 25,000 new cases of breast cancer in 2020 alone. And 1 out of 9 women in Pakistan is at the risk of developing breast cancer. The global mortality rate is equally concerning, with approximately 0.6 million casualties each year. So breast cancer is caused when the cells in the breast tissue grow uncontrollably due to malfunctions and abnormalities in the signaling pathways. ESR1, which is the estrogen receptor, is the most crucial one in causing breast cancer and the endocrine resistance. When activated by estrogen, this receptor causes mutation in itself and causes an automated expression of itself and then leads to endocrine resistance, and the cancer cells become less responsive to the normal therapy, chemotherapies and hormonal therapies. So this is the major challenge that we are facing these days in terms of treating cancer. Estrogen receptor is a 595 amino acid protein with 6 functional domains, and the ligand binding domain of this receptor is the most important in causing endocrine resistance. So there are a lot of mutations already been reported in literature in the ligand binding domain, but the most important one in causing this sort of resistance is D538G. This is a substitution and missense mutation, which means that at 538 position, the amino acid aspartic acid is being converted into glycine, and it is causing a frame shift, and the whole structure of the receptor is being changed. So the problem that we are addressing here is that the current therapies of breast cancer have limitations due to mutations in the receptor. Therefore, it is important to study the prevalence of this hotspot mutation. And it is also important to identify new drugs targeting the mutated receptors and offering alternative treatment options for endocrine resistant breast cancer. This is a brief overview of our methodology. We collected breast cancer samples from 3 different hospitals, and we extracted the genomic DNA and checked the prevalence of this resistance causing mutation in our samples. Then we sent it for next generation sequencing over to South Korea. And at this stage, we were astonished to observe new mutations being detected in the gene of Pakistan. Okay. So after the sequencing and detection of new mutations, we performed in silico analysis, and we purposed drugs against.
Aruj, we can hear you, but can you please share your slides as well?
Sure, I can share my slides.
And you
Just
can
give-
start now.
Okay. I hope my screen is visible to you.
Yes, we
Hello.
can see. We can.
Okay. All right, I'm going to start now. Assalamu alaikum and good afternoon, everyone. I'm going to talk about the role of RALA and RALP proteins, their structural insight, and their membrane dynamics. I'm Dr. Aruj Shafiq, assistant professor at Aga Khan University, Karachi. So RAL proteins are the GTPase family members. These they cycle between active GTP and inactive GDP bound forms. These proteins this pathway is the third best characterized pathway downstream KRAS after PI3 kinase and MAP kinase pathway, and its drug target from last 2 decades. Many inhibitors of this pathway are in clinical trials now. These proteins, despite their huge sequence and structural similarity, tend to play a differential role in many cancers. For instance, RALA has been shown to more role into encourage-independent growth and tumorigenesis, while RALB has more role into cell survival and cell mobility. The G domain of these proteins is of higher sequence similarity, and many structures of this G domain are available. However, there is a 2025 residue hypervariable C-terminal region. This region has low sequence similarity between these 2 proteins. It has predicted helix in RALA, but not in RALB. This region undergoes differential phosphorylation as well. And it also has been shown that in vivo study, it binds to the calmodulin in a calcium-dependent manner. And the C-terminal CAX motif, the very end region undergoes cleavage, and then it undergoes lipid modification, which enables these proteins to anchor to the cytoplasmic side of the membrane. So what the gap here is, so we don't have a structural information into this hypervariable membrane bound region, and we have a very little in vitro information regarding binding of the RAL protein with calmodulin. So the objective of this study were to actually look at the expression of these proteins into RAL in the colorectal cancer patients, and also to look at the structural dynamics of the G protein. So we do know the structure, but is there any dynamic changes differences between RALA and RALB which drives their differential function? So we were looking at that. And the third objective was to actually characterize the RAL binding to the calmodulin, calcium calmodulin. So the methodology we used for the expression study, we used publicly available TCGA cancer transcriptomics datasets to look at the expression of these proteins in cancer or colorectal cancer patients and normal tissue samples. We used NMR to solve the structure of the G domains and to look at the structural dynamics of the G domain. And we also looked at the NMR to actually look at the regions of the RAL-A and the calmodulin, which are involved in the binding and the interaction between these two proteins. The binding affinities between the RAL-A, RAL-B, and calmodulin were determined using surface plasmoresonance imaging method. This method is actually based that if we actually bind one of the protein into the surface, and we actually the other protein is in the mobile phase, and if there is any binding happens, the angle of reflection of this light will change. And the stronger the binding is, the more change in the in this reflection angle will be observed. And using this, we were able to determine the KD values or the binding affinities between calmodulin and RAL proteins. So let's look at the results. In terms of expression studies, when we did look at the expression of RAL-A in the normal versus tumor sample, there was a significant difference between the expression levels. And we did observe a difference and an impact on the survival of the patients. With the higher expression of RAL-A, there is a lower survival in the colorectal cancer patients. On the other hand, we didn't observe any significant difference between the tumor and the patient samples, normal tissue samples in RAL-B expression, and there was no difference impact on the higher expression on the patient survival as well. So then we look at the dynamics of the G protein. Here, these two structure actually represents the active site dynamics, active form of the proteins. And here, these left on the very right, these structures represent the inactive forms of the protein. Overall, if we combine this analysis where the colors are actually representing the differential dynamic properties or the binding properties at nano and microsecond scales, we observe that the major difference lies in this loop region, which is 116 to 121 amino acids. There is an insertion of one residue in RAL-B, and the dynamic differences of this loop region actually also affects the adjacent helix region, which is also has a different orientation in between these two proteins. Other major difference we observe in the switch region, but this is expected because the switch regions are binding to the nucleotide. So this is an expected difference in the dynamic properties, but this difference in the loop region might actually be responsible for some of the functional differences between these two proteins, and it needs further in vivo studies to investigate that. So then a third objective of our study was to actually look at the RAL and calcium calmodulin binding. This is of a huge significance because this interaction is of drug target even in case of KRAS, because KRAS also binds to the calmodulin, and there is a very little structural insight available for the structure-based drug designing. So what we did in the first, we look at the binding affinities. So what binding affinities, when we were determining the binding affinity, we found that RAL-A actually binds with a stronger binding affinity with a KD value of 45 nanomolar compared to the RAL-B, which has a KD value of 95 nanomolar. And then we actually look at the interactions, so which regions of RAL-A are involved in the interaction. The method of doing was that we isotopically labeled RAL-A, and then we titrated in calmodulin. So the regions which are actually involved in the binding, the peaks will shift. And what we observed is that the peaks of only this C terminus, the very end region, which is hypervariable region in RAL-A, was shifting. So that means only the RAL-A C terminus is involved in binding to the calmodulin. On the other hand, when we actually labeled calmodulin and we titrated in RAL-A, what we observed that there were the region across the whole protein which were actually whose peaks were shifting. So that means a larger part of this calmodulin protein is involved in RAL-A binding. And then we used the RAL-A C-terminal helix bound to its lipid modification. And we saw the structure of calmodulin. Here, this white lobe region actually represents the two lobes of calmodulin protein. And this pink region is actually the RAL-A C-terminal helix bound to this protein. So this does show that both loops of this calmodulin proteins are actually involved in the interaction with RAL-A. This interaction and this structure is of huge significance because it gives us the first insight into the structure of this kind of interaction, and this interaction is this can actually be the base to develop the structure based drugs to target this interaction in cancers, not only for RAL-A and RAL-B but also for KRAS, which has a very similar C-terminal region, and the residues which are involved in interaction are of the same nature between RAL-A and KRAS. So in the end, I would like to thank my supervisor, Dr. Helen, at University of Cambridge, my colleagues at University of Cambridge. We published this work in PNAS, which has an impact factor of 12. And at Aga Khan University, I'm actually carrying forward this work in patient samples in our own population, where Miss Nazia, which is my MPhil student, and Miss Taira, my RA, are working with me on this project. Thank you very much. And any questions?
Thank you, Aruj. It was a very interesting talk, actually. I think there is somebody in the audience who would like to ask a question. Yes, please.
Should I stop sharing my screen?
Yeah, that's fine.
Oh,
Thank you.
Hello, Aruj. Assalamu alaikum. How are you?
Walaikum. I'm good. Alhamdulillah.
Can you talked about, you tell us about the effect of phosphorylation on calmodulin binding and other things?
All right. Can you hear me? Assalamu alaikum. How are you, sir?
Good, thank you.
All right. So good to see you. Actually, we, in terms of phosphorylation, we did observe some changes. I didn't show that data, but the KD values were changing. I didn't include that data, but when the phosphorylation and dephosphorylation state, the KD values were changing. And that is also one of the reasons for the difference in the KD value between RAL-A and RAL-B, because they have a different phosphorylation site at the C-terminus.
Okay, thank you.
Okay, Aruj, just one more question from me. You said that KRAS also binds to calcium calmodulin.
Yes.
So, yeah, so if there is a mutation in KRAS, naturally it will affect the binding. So you are
No.
looking, yeah. Okay.
Sorry, please continue.
You carry on, yes, so you wanted
Okay.
to say.
So most of the mutation in the KRAS are in the G domain regions, which actually impact the their activity or their activation in cancer. The C terminus regions, they have a very little information in terms of structure, but there is no C terminal mutation which is known to be involved in cancers. So the binding is actually at the C terminus, so there is no known mutation in cancer at the C terminus.
Okay, so we are good to go with new drug discovery for that.
Yes, yes.
We know those 4 classes of neuronal sodium channel blockers. Can you name some drugs?
Well, I don't know the specifics, but the research that arrived at the conclusion that sodium channel blockers might work on the SCLC patients did use several of the neuronal sodium channel blockers, plus they used many other drugs that basically was inhibiting the neuronal activity in those SCLC cells.
All right. Thank you so much. Best of luck for your research.
Thank you. Thank you.
Faizan Fazal from Benazir Bhutto Hospital, and he'll be talking about the effect of adding radiotherapy to androgen deprivation therapy in men with node-positive prostate cancer after radical prostatectomy, a systemic review and meta-analysis. Next is Dr. Akil Shahid, Fellow Clinical and Radiation Oncology at SKMCH Lahore, and he'll be talking about clinical and dosimetric outcomes of craniospinal radiation using rapid arc technique in a low and middle income countries, a single institution experience.
So, Assalamu alaikum and good evening, everyone. My name is Dr. Akil Shahid, and I'm a fellow clinical and radiation oncology at SKM Lahore. and I will be presenting my lecture on the craniospinal irradiation. So starting with the definition, what is actually craniospinal irradiation? So when we actually treat the whole of the cranium and the spinal axis, this technique is basically labeled as the craniospinal irradiation. Now briefly touching the epidemiology. So primary CNS malignancies comprise of almost 20% of all cancers worldwide, with over 250,000 deaths reported. In Pakistan, over 5,000 cases were reported in the year 2022, with over 4,000 deaths. And to narrow down the epidemiology, in specifically pediatric population, the most common type of cancer is medulloblastoma, with around 23.7% prevalence. So why this stat is important? Because treatment of cancers such as medulloblastoma, ependymoma, and germ cell tumors. These are the tumors which actually tend to spread through the cerebrospinal fluid, and these are the tumors which actually necessitate the treatment of the whole neural axis with craniospinal irradiation. Coming to the brief evolution of how actually CSI has evolved, because this is part of because it is more important to understand the CSI technique itself in order to actually grasp the results of our study. The craniospinal radiation was first basically reported in Acta Radiologica in 1953. The first advanced technique was actually developed by Dr. Edith Patterson, and the study showed that conducted on 27 patients showed 3-year survival of about 65%. This was the first technique which was used for craniospinal radiation. You can see that there are 2 spinal fields and 2 lateral fields for the cranium. And this was the conventional 2D technique. We did not know what actual doses was being given to the spinal cord or the brain, because we did not have any modalities to actually measure the dose which was being given into the body. So we actually just applied the fields and calculated the MUs and operated the machine such. So no actually dose volume could be calculated. So now coming to when we had 3D scanners introduced and the technique involved with 3D conformal technique, I would like you to focus on the red, yellow, and green parts. The blue part is actually the low dose region, and we're not very much concerned about that. So you can see that this is the red part is actually the dose which was prescribed, and you can see that clearly the dose prescribed was exceeding that from the spinal cord. The yellow being the high dose region and green being fairly less, but the yellow and the green were also covering the normal tissues which were not part of the disease, thus increasing the toxicities in this patient. As the technique evolved, we came to the era of IMRT, and you can clearly see that we had actually limited the high dose region to basically the spinal cord, but clearly there is no homogeneity in the dose. There is quite an unevenness throughout the spinal cord and throughout the doses. So again, this required further enhanced technology to basically minimize the toxicities. And now coming to the VMAT technique, the currently used modality, and you can clearly see that there is a very homogeneous dose throughout the spinal cord, with high dose region clearly focusing on the spine itself, and the low dose region, the overall organs which are not involved are clearly under the low dose region. So we have a very good toxicity profile with this technique. So with VMAT technique, there is a significant reduction in doses to the organs at risk. These are the organs which are not involved by the disease, but do come under the radiation field. And these need to be actually mindful. We need to be mindful of these, because these are the organs which actually cause the toxicity. And thus, this technique actually improves the quality of life in this patient. Now coming to my study, which is the clinical and dosimetric outcomes of craniospinal irradiation using rapid arc or VMAT technique in a low middle income country. And this is a single institute experience. The objective of my study was to analyze the clinical and dosimetric outcomes using rapid RVMEC technique for CMS tumors treated with craniospinal irradiation at Shaukat Khanum Hospital, Lahore. This was a retrospective descriptive analysis. All the patients which were treated in the past 4 years were taken. Who were treated with craniospinal radiation was taken. Their data was collected through the hospital information system and was analyzed through SPSS. So now coming to results, a total of 23 patients were treated with a mean age of 13 years. The majority of our cases were actually medulloblastoma, and 21 patients out of 23 had grade 4 tumors. Out of these medulloblastomas, 12 were classified as high risk, while CSF was positive in 20 patients. Surgical resection was carried out in all the patients, while 11% of the 11 patients had complete surgical resection. Concurrent chemo was given in 15 patients, while with this technique, as I showed you the development, only 17 patients had minor neurological toxicities. 3 patients had absolutely no neurological toxicities, and only 3 patients developed some major form of neurological toxicities. And we experienced recurrence in only 4 patients. So the doses used were fairly standard. These are the doses which are used worldwide for standard risk. The whole of the craniospinal axis was treated with 23.4 gray, while for high risk, 36 gray was given to the craniospinal axis. And the boost dose, this is given to the posterior fossa. This was between the range of 45 to 55.8, depending upon the proximity of the lesion itself and respecting the organs of risk and their tolerances. And all doses were given with 1.8 gray per fraction. This is the acceptable fraction because in especially pediatric population. So all our patients were treated using 2 full arcs and 4 partial arcs for spinal coverage. The mean PTV 95% of coverage of the craniospinal irradiation, the craniospinal axis, was 99.1%, while the mean PTV 95% coverage for the boost or the posterior fossa was 98.7%. Coming to the dosimetric analysis, these are all the organs which are at risk during treatment with craniospinal irradiation. And you can clearly see that none of our organs at risk exceeded their tolerances doses in any of our patients. So to conclude, craniospinal radiation using rapid arc VMEC technique is currently the most effective way of delivering such a complex treatment plan. All our doses were within their tolerances, and we had very favorable outcomes with our patients, and our results were very much comparable with the rest of the world. Coming to the future directive, so I just wanted to, I tried to explain you the techniques. So this is the latest technique. This is the proton beam, which is now being currently implemented. And you can see there is absolutely no dose fall off anywhere else on the body. All the prescribed dose is very well confined to the spine, and this is the way forward. I thank you all for the talk. Any questions would be appreciated.
Thank you. Yep. So just a quick, is there any question from the audience? Just a quick question. Have you compared your results with the previous patients that you have treated at your institution with 3D conformal radiotherapy in terms of toxicity and outcomes?
No, we haven't, because since the start of this technique since it was introduced, we have treated all the patients with the same technique. We haven't found any patients who were treated with 3D conformal.
Okay. But maybe it's worth looking at the data for those patients who were treated before rapid arc with 3D and comparing the toxicities at what
we
would
can
have
certainly
changed
compare
and
that.
looking at their doses. Any other questions? Thank you very much, Akhil.
Thank you.
So next, I would like to invite Dr. Saira Siddique from Kiran Hospital, Karachi, and she'll be talking about dosimetric feasibility and acute side effects with high dose rate CT-guided 3D dimensional brachytherapy in carcinoma of the cervix experience at Noreen Hospital.
Online.
No, she's not there online as well. So we are moving forwards quickly now. Can I have request Dr. Anusha Haider, radiation oncology resident at Shaukat Khanum Memorial Hospital, Lahore, and Anusha will be talking about endometrial cancers and emerging perspective on therapeutic strategies.
Assalamu alaikum, everyone. I'm Anusha Haider, a second-year resident in clinical radiation oncology from Shaukat Khanum Cancer Hospital, Lahore. Today, we'll be discussing the emerging perspectives on therapeutic strategies in terms of endometrial carcinoma. We'll be focusing on the epidemiology and risk factors, the histology, the cancer genome atlas molecular classification, the adjuvant treatment paradigm, the center-based experience. So it's one of the most commonly diagnosed gynecological malignancies worldwide. The first symptom being the pervaginal bleeding leads to an early detection and thereby the treatment that has led to an astonishing decrease in the number of deaths worldwide to a level of 2.2%. The risk factors are further categorized into two: sporadic, making up 90% of cases, followed by the hereditary, which makes up around 5 to 10% of cases. The most common factor in terms of sporadic endometrial carcinoma is the exposure to estrogen, followed by obesity or high BMI. In terms of hereditary factor, the most common is the Lynch, which is an autosomal dominant genetic condition causing one of the contributory factor in causing endometrial carcinoma. If we talk about the histology, the World Health Organization has classified endometrial carcinoma into the following subtypes. The endometrioid makes up the most common subset of these, followed by serous and clear cell. The remaining undifferentiated, dedifferentiated, mixed carcinosarcomas, they make up the relatively small percent of all cancers. So in 1983, Bachman classified endometrial carcinomas into two main categories, type 1 and type 2. So type 1 makes up around 80% of all cancers, are estrogen-driven and cause endometrial hyperplasia. The most common histology associated with them is the endometrioid, and they're generally low grade. And fortunately, they do carry a favorable prognosis. Comparing type 1 to type 2, the type 2 make up around 10 to 20% of all cancers and are not estrogen-driven, arise from the atrophic endometrium and carry serous and clear cell as the most common histology, are high grade and unfortunately have high relapse rate and unfavorable prognosis. But this classification lacks reproducibility in terms of practical implications of endometrial cancer. So the Cancer Genome Atlas proposed a recent classification following and focusing on the 4 molecular subtypes, and this involves sequencing. So sequencing is a cumbersome process to actually carry out. These surrogate markers can also be used for making a TCGA-inspired molecular classification. It simply means that any endometrium pathology specimen that you have, we need to run 4 testing on it. The first is a POL-E mutant, POL-E status testing. So those which test positive for the mutation will be labeled as POL-E mutant carcinomas. Those which are proficient in them will be tested for the MMR status. So loss of any of the 4 proteins, the MLH1, MSH2, MSH6, or PMS2, deficiency of any one of these 4 will label the person as a deficient MMR endometrial carcinoma. And those which are not deficient in any of these will be labeled as a proficient, and they will be further tested for the P53 status. So those which are wild type will be labeled as no specific molecular profile, and those which are mutant will be labeled as P53 mutant endometrial carcinoma. So the POL mutant carcinomas generally have a favorable prognosis, but the mutant, they carry the worst prognosis. The deficient MMR and the no specific molecular profile, they carry an intermediate prognosis that is definitely stage dependent. So we talk about the adjuvant treatment paradigm. We classify them as low risk, the intermediate risk, the intermediate high risk, and the high risk category. So the low risk patients will be those which are stage 1A, grade 1 and grade 2 with no or focal LVSI. Now, I'd like to mention what I mean by focal LVSI. So it is the tumor involvement in less than 4 vessels. So in these patients, generally adjuvant therapy is not recommended. Another thing to note is a stage 1 and 2 pole mutant cancer. And in these patients as well, due to favorable prognosis, adjuvant treatment is something which can be skipped. So we talk about the intermediate risk factors, stage 1A, grade 3, with no focal LVSI, deficient MMR, or no specific profile are those in which vaginal brachytherapy is recommended alone. It's the same as for stage 1B, grade 1 and 2, stage 2, grade 1, with no focal LVSI also require vaginal brachy alone. For patients which are stage 1A, P53 aberrant, not infiltrating the myometrium, adjuvant treatment is also not indicated. So if we talk about the intermediate risk, high intermediate risk and the high risk, these are the patients which will be requiring external beam radiotherapy, including stage 1A and 1B with substantial LVSI. Now that is what will be involving more than 4 vessels and includes stage 1B, grade 3, stage 2, grade 1, stage 2, grade 2, and grade 3. In terms of the high risk, all the pathologies which are P53 aberrant and those which are serous and clear cell carcinomas will be the patients which will be requiring aggressive treatment with chemoradiotherapy given with cisplatin 50 milligram per square meter. So the adjuvant treatment, as discussed earlier, requires external beam, chemo, or vaginal brachytherapy. So in terms of carcinosarcomas, there is continuous testing has been proposed widely because of the improved progression-free survival and overall survival in terms of using trastuzumab. The world-famous PROTECT-3 trial shows progression-free survival and overall survival in patients which are treated with chemoradiotherapy in comparison to pelvic therapy alone. So is there a role of immune checkpoint inhibitors? There is a definitive role of immune checkpoint inhibitors when talking about the patients who have advanced or metastatic endometrial carcinoma. That is evident by the RUBY trial and the NRGGY018 trial. Dostarlimab is an immune checkpoint inhibitor that showed progression-free survival in patients in the deficient MMR group. In comparison to the NRG uses pembrolizumab, which showed progression-free survival and overall survival in patients which were both deficient in MMR and those which were proficient in MMR. So keeping in view the above discussion, we evaluated around 121 patients in our center. Unfortunately, POLE-E mutation is not being tested anywhere in Pakistan, and the MSI testing in our center was around very low as well. So there was only one patient who was tested for it. In terms of P53 mutation, there were around half of the patients which were tested for P53 status, and they showed that around 38% were wild type and 6% showed mutant pathology, and those were definitely which required aggressive treatment. So the main idea of the whole discussion was to incorporate the molecular testing profile in our treatment paradigm, so to stratify the patients and decreasing the incidences of over and under treatment. And it also reduces the unnecessary side effects and toxicity experienced by the patient, and also explores targeted medicine, including checkpoint inhibitors, and it definitely decreases the cost-related burden on the institute. Thank you very much. I'll specifically like to mention my colleague, Dr. Anis, for his immense help in the presentation and everything. If I have any questions, I'd be happy to answer.
Thank you, Anusha. Yeah, there is one question in the floor. Can you give the mic, please?
Thank you so much for the wonderful talk. I would like to ask if the POLE-E mutation that you mentioned in your presentation, is it available in Pakistan?
So unfortunately, it's not available in Pakistan in most of the centers, but it's something which should be carried out for all biopsy specimens. So if you don't have it, you can go for the MSI testing and for the P53 status testing.
Sorry, can I ask?
Yes, sure.
Your P53 mutations, where did you perform that?
Yes, they were performed at our institute.
In our institute?
G.
So
Yes.
you did the cancer hotspot panel, and from there you got?
Yes.
Okay. So you just wait, POLE is coming too quickly.
We'll be happy about that. Thank you so much.
Okay, now I would like to invite Furqanul Haq, SKMC, Peshawar, Pakistan. He'll be talking about Battle of the Protocols, assessing the effectiveness of cross versus flot in esophageal adenocarcinoma.
Assalamu alaikum. Good afternoon. My name is Dr. Furqanul Haq. I am working as a resident radiation oncology in Shaukat Khanum, Peshawar. Today, I am going to present the battle of protocols cross versus flot in in the treatment of esophageal adenocarcinoma. Esophageal cancer, basically epithelially, there are two main histology present in epithelium of esophagus. In upper two third, there is a squamous cell epithelium, and it leads to squamous cell carcinoma with patients who are smoking, and these are more prevalent in people of Europe and Asia. Adenocarcinoma, they are more commonly developed in the lower third of the esophagus and originate from Barrett esophagus. And adenocarcinoma is most commonly present in America and Europe due to increase in incidence and prevalence of obesity and alcoholism. The treatment approach in locally advanced gastroesophageal junction adenocarcinoma. The optimal treatment strategy for locally advanced distal esophageal and gastroesophageal junction adenocarcinoma is under debate. Surgery as a main curative modality is usually combined with either perioperative chemoradiation or perioperative chemotherapy followed by surgery. The learning objectives are to compare and see the results of treatment who have in patients who have esophageal adenocarcinoma and gastroesophageal junction adenocarcinoma in our center. The sample size of the study, which were done from 2019 to 2024 for cross trial, were 18, in Florida were 38. So moving forward to the cross trial, the beginning of neoadjuvant chemoradiation therapy before surgery. So we can look at the graph and see that the arm of patient who have been treated with chemoradiation therapy followed by surgery are having better proportion of surviving as compared to the surgery alone. Then moving forward to the MAGIC trial, and in which the patient have received pre-op chemotherapy ECF regimen, 3 cycles 2 weekly for 3 cycles, followed by surgery, followed by 3 cycles 2 weekly in MAGIC trial. The ECF stand for epirubicin, cisplatin, and 5-fluorouracil. So we can look at the Kaplan-Meier analysis and see that the progression-free survival in the pre-operative chemotherapy followed by surgery were better in the group as compared to the surgery alone group. And the overall survival were also better. So now moving forward to the most popular trial in esophageal and gastric adenocarcinoma, the FLOT4 trial, which basically they have compared the two regimen, the ACF regimen and the FLOT regimen. So the ECF, and they found that the overall survival in the FLOT group were 5-year overall survival were 45%, which is quite more than the ECF regimen. But there was no trial to compare the chemoradiation therapy before surgery with the pre-operative chemotherapy alone. So it was the New Ages trial which answered the question that whether the chemoradiation therapy before surgery is better or the only chemotherapy group. So they took two arms. In arm A, they took the MAGIC regimen and the FLOT regimen. And arm B, they took the cross trial or the cross protocol people. We can look at the table and we can see that the R0 resection with the MAGIC trial were 82%, in arm B or the cross trial they were 95%. The nodal regression was 44%, and in the arm B the cross trial which was 60%. And the tumor regression were 41% in the cross trial as compared to the arm A or the FLOT or MAGIC trial, which were the 12%. So by looking at the New Ages trial, we concluded that the neoadjuvant chemoradiation followed by surgery became the standard of care in Sievert 1 and Sievert 2 gastroesophageal junction tumors. For Sievert type 3, which has been classified into TNM 8 and 9 as a gastric tumors, and those have been treated with FLOT regimen. So now, after the new age trial, there came a new trial which is still ongoing. We call it ISOPEC, and they supported the FLOT regimen. And if they followed the patient for 6 to 7 years, and they found that the 3 years overall survival, which is 57.4% as compared to the 3 years overall survival of the patient who have been treated with the cross regimen, which is 50.7. And the in 5 years overall survival or 50.6% as compared to the patients which were being treated with the cross protocol, which is 38.7%. So the debate was going on, and we wanted to look at our patients and see the responses in our patient. So this is our pilot study outcomes in our patient treated in Shaukat Khanum. Now, the pathological complete response rate, we found that in the chemoradiation therapies of 5.5% as compared to the FLOT regimen, which is 5.2%. The planned treatment completed was in chemoradiation therapy group 92% as compared to the FLOT, which is 80%. The rate of tumor regressions, we compared the pre-treatment stages to post-treatment pathological staging, and we found that with chemoradiation therapy, the tumor regression was 53% as compared to the FLOT regimen, which is 52%. The rate of bone marrow suppression, basically we look into the neutropenia, and which is more in the chemoradiation therapy group. And those were 40% in chemoradiation therapy as compared to the FLOT regimen, which is 32%. And we found that we are using modified cross protocol, and we give neoadjuvant 2 cycles of pegcilcarbo 3 weekly before the start of chemoradiation therapy. So that's why all patients who have been treated with chemoradiation therapy develop greater rate of bone marrow suppression as compared to the FLOT regimen. The rate of hospitalization were more in the FLOT regimen as compared to the chemoradiation therapy. And the rate of GI side effects were also more found in the FLOT group as compared to the chemoradiation therapy group. So this is the summary of, I have made a table for that, and we can see that the pathological response rate and tumor regression, the gastrointestinal side effects, planned treatment completed, and rate of hospitalization and neutropenias were better in the cross group as compared to the FLOT group. So we conclude that the chemoradiation therapy is better modality of treatment in CVERT1 and CVERT2 esophageal esophageal adenocarcinoma and gastroesophageal junction adenocarcinoma. So my future goal is we want to do a randomized control trial to prove which one is better to set the standard of treatment of choice for esophageal adenocancer for our patients who have been present in Pakistan, Afghanistan, and the areas around. I would like to thank everyone, and please ask any question if you people have. Thank you so much.
Thank you, Furqan. It was a wonderful topic. Are there any questions from the audience? Can I just ask one question? How do you define which patient should be getting perioperative chemotherapy with FLOT versus chemoradiotherapy? So how that decision is made upfront?
Thank you so much, madam. Basically, we have to select the patient. First, the patient have to have adenocarcinoma of esophagus. Second, the patient need to have a T2 or more than a staging T3 or nodal positive disease to select the patient for to get the neoadjuvant chemoradiation therapy or the neoadjuvant chemotherapy.
So that is what my question is. Once you have decided that this patient needs neoadjuvant treatment, which patient should get concurrent chemoradiotherapy versus FLOT? How do you decide between these two arms?
Thank you so much, madam. As far as we can look at the results of our pilot study, the result shows that we need to give chemoradiation therapy as a new adjuvant chemotherapy, new adjuvant treatment in esophageal adenocarcinoma. But if the patient is having severe type 3, then we will select FLOT regimen. As because the severe type 3 has been classified according to TNM8 and TNM9 stage as a gastric adenocarcinomas, and it shall be treated with FLOT regimen just like gastric adenocarcinoma.
Thank you very much. So that means both the populations are not interchangeable. So there's a different population which will get chemoradiotherapy, and there's a different group of patients with severe type 3 which will be going for FLOT therapy. Thank you so much. Thank you.
Yes, thank you so much, madam. Thank you.
So moving ahead, the second last topic of talk of our session, I'd like to request Momina Mustafa from SKMCH Peshawar. Is Momina here? Is she online? No. So the last talk for this session is by Dr. Isha Khalid. Isha is a radiation oncology resident at SKMCH Lahore, and she'll be talking about LINAC-based stereotactic body radiotherapy, a single institution experience.
Assalamu alaikum. I'm Isha Khalid from Radiation Oncology Department, and today I'm going to present my talk that is the NAC-based stereotactic body radiotherapy, that is SBRT, in low and middle income countries' institutional experience. So before I start, I would like to familiarize you with what SBRT is and what do I mean by LINAC-based SBRT. So it's an advanced radiotherapy technique where we treat the tumor with a high dose to a very small, precise target, along with minimizing the healthy tissue damage. So linear accelerator, or LINAC, it's the radiotherapy machine where accelerated electrons are used to produce high energy photons that is used to deliver highly conformal radiotherapy. That's why we have effective treatment outcomes and fewer side effects. LINAC-based SBRT was first introduced in 1995 by the team led by Dr. Robert Timmerman. And here you can see the different lesions. With advanced radiotherapy technique, we can offer excellent tumor control as compared to surgery, as evident from the literature that it ranges from 82% to 97%. So SBRT it's a complex process. It involves multiple steps, starting from simulation, in which a CT scan is done with appropriate immobilization and motion management, especially for tumors which have physiological movements. For example, liver or lung lesion that moves with respiration, we have to perform scan with special motion management. And then this is followed by contouring and planning part, which is done by oncologists and physics teams. And then they both sit together and finalize the plan. And then the whole radiotherapy treatment is delivered to the patient. So this all combines to give that highly conformal radiotherapy, which is our aim. But this needs high upfront costs, advanced imaging and treatment planning systems, and we need highly trained professionals for it. So this is a challenge for a developing country which has limited resources, especially in an institution like ours, where we have to provide this kind of treatment free of cost. So the purpose to conduct this study was to assess the outcomes of the patients that were treated with SBRT, and to see the quality of treatment plans, as are we meeting the international standards. So we recruited patients that were treated with SBRT from 2021 to 2023 at SKM Lahore. We included all those patients with extracranial lesions that were treated with SBRT after MDT discussion. Outcomes included overall survival, progression-free survival, local control that were analyzed using Kaplan-Meier curve. The treatment plan quality parameters were presented as mean and standard deviation. The qualitative variables were presented as percentages, and this is all analyzed using SPSS software. So total 54 patients were treated with median age of 49 years, and predominantly there were females. Here's the list of lesions that were treated, and you can see that 50% of the patients had spinal or bone disease, followed by liver and nodal, followed by prostate and adrenal lesions, followed by lung. And the dose ranged from 16 gray to 54 gray in 1, 3, or 5 fractions.
So the outcomes, 2-year overall survival was 85.8%, 2-year progression-free survival was 74.9%, and the most important one, 2-year local control was 94.3%. And this is comparable to the data published by international countries. So this is comparison of local control, site-wise local control in our current study and the literature, and you can see they are highly comparable. As far as response is concerned, we got 18% radiological complete response, and 63% of the patients had stable disease, and there was progression in only 3 lesions. 2 of these lesions had rectal adenocarcinoma as primary, and 1 of these lesions had malignant melanoma as histology. Coming to the treatment plan quality, the mean CTV coverage, that is the clinical target volume coverage we obtained, was 98.9% with standard deviation of 1.73. And let me explain you what do I mean by CTV coverage. It means that our 99% of our volume should receive the prescribed dose near to 100%, and we are near to 100%, that is 98.9%. And the other is the planning target volume that includes CTV plus the surrounding setup margin, and we aim to be D99%, more than 90%, and we achieved mean coverage of 95.6% with standard deviation of 4.93. Mean RTOG conformity index, that shows that how well our plan is, our radiotherapy dose is confined and conforming the target, that should be ideally be 1, and we achieved mean of 1.01 with standard deviation of 0.06. So organ at risk dose constraints were achieved in 100% of the plans. Here I present 2 of our cases of SBRT. On the right hand side is a lung lesion, and you can see that the dose is highly confined within the PTV volume, and the maximum dose is in the center, and there is minimum splash in the surrounding healthy tissue. On the other side, there is a spine SBRT case in which we aim to give 100% dose to the vertebra, but we have to save the central spinal canal. And this is a typical donut-shaped color wash in which the dose is confined to the vertebrae, and there is minimal splash to the central spinal canal. So in conclusion, LINAC-based SBRT can effectively deliver ablative doses to the target while sparing the organ at risk, and offers excellent local control. So in developing countries like ours, where we have limited lines of treatment, and we will have SBRT, an excellent alternative that can provide excellent local control and comparable outcomes. These are the references. Thank you so much. Any questions? Yes, it is site dependent, as I have shown 2 of the cases. One is the lung one. We treated it with 3 non-coplanar arcs, and the spinal one we treated with 2 full arcs.
So what is your, I think I missed that point where you mentioned about your mean biological equivalent dose. So what was your mean biological equivalent dose in single fraction, 3 fractions, and 5 fractions?
I have assessed the treatment plan quality parameters using mean PTV, CTV coverage, that is D99%, and the conformity index, by which I am defining the plan quality parameters.
We have not calculated our BEDs.
No, we have not calculated the BEDs.
Thank you very much, India. Thank you.
Okay, thank you. Now is the last talk. Sorry, it was missed in the printing. I will invite Dr. Humaira from SKMCH Lahore. She'll be talking on PCR RFLP methodology, a cost-effective solution for rapid molecular identification of Candida species in LMICs. Thank you, Humaira.
Assalamu alaikum, and thank you for everyone for being here with me to listen. Although all the chicken is gone, but I think few people are still here and I can offer the nuts for you. My name is Dr. Humaira Farooq, and I'm a clinical scientist for fungal infections in Shaukat Khanum Memorial Cancer Hospital. And a little introduction about me that I have recently, almost 1 year back, I finished my PhD, and now we are trying to establish first reference medical and molecular biology lab in, it is a first in, I think it is a first in Pakistan, and we are under the roof of, we are trying to establish under the roof of Shaukat Khanum Memorial Cancer Hospital. So, a little background of the study. First of all, the title of the study is, I am here to present the results of my recent study, which is just finished and is in the process of publication now. And it is in the series of many other studies and following the same goals as I have mentioned, I have discussed with you before. So, little background of the study that WHO almost estimated 1.7 million people die every year globally because of the fungal infections, but because these infections generally are not reporting properly, so it is very easy to sweep the fungal infections under the rug. So there is a constant increase in fungal infections and their resistance to the currently available medicines. And we need to keep our eyes open because before that, many emergencies have occurred, especially in India for fungal black fungus emergency. So new fungal pathogens are also emerging, and they have the great potentials to cause such medical emergencies like the black fungus globally. Despite all these factors, medical mycology is still neglected area globally, including Pakistan. So keeping in mind, we designed our study with the main objectives to develop cost-effective, sensitive, accurate, and DNA-based molecular identification methods. That can fill also the epidemiological gaps by estimating the rate of yeast infections and also the emerging infections in tertiary care hospital. The study this study was a cross-sectional study, and that was designed basically for 6 months duration, and sample size was calculated about 120 samples, and the samples were collected from the microbiology lab of Shaukat Khanum Cancer Hospital, while the data was collected from the microbiology and the hospital information system. Little about methods. As I have mentioned in the title, our methodology for molecular identification was the PCR to RFLP. And RFLP basically, it is a restriction fragment length polymorphism. Basically, this is based on the cuts of the specific DNA by restriction enzymes. This is basically restriction enzymes based methodology. Sporting methods were microscopy and chromogenic agar media. Few people who don't know about the restriction fragment length polymorphism, that what is it? Here I just a few clicks about, tell you about this method. Actually, it is a method that is used to identify different or types or the forms of an organism. It involves cutting of the specific DNA of different species using restriction enzyme, which can cut the specific DNA into fragments of different sizes. And the differences in the sizes of these species can help distinguish between different species or organisms. For example, you can see here that restriction enzyme, they are the three kind of different species of the same organism, but you can see in line 1, the restriction enzyme did not digest it or cut the DNA. But in the second type, it is cut the DNA in 3 fragments. In the third type, it is cut the DNA in 2 fragments. And variations among these fragments is the key to for this methodology that you can distinguish between the different species. A simplified flow chart of PCR of this method you can see here. First, samples were collected from microbiology lab. The DNA was extracted from them, and then specific DNA was amplified using PCR. And after that, the PCR products were cut with the restrictions, specific restriction enzyme, and visualized, and the patterns were studied. Basically, in this methodology, major very basic or very important part is to select the enzyme that can cut the DNA in such a way that you can distinguish between different species. You need to select the best enzyme. That is the major part in the, the studies which are, they are basically depend on the selection of the restriction enzyme. In this process, you need to obtain the specific DNA sequences of the species and then paste them into the different databases, DNA databases like NABCutter in European data. It is a European database, and some people use Japanese database, DNases. And after that, you get the enzymes and the best results, the enzyme which show the best results, you have to choose it for your study or for your, experiments. This study was basically inspired and used the publications of Iranian medical mycologist, Professor Dr. Hussain Mirhandi, that was my field advisor also for this work. And here you can see that different species of the Candida showing the different patterns of DNA bands. So you can identify by these patterns different species. These are my results. The microscopic view of the different species of Candida, you can see here that many, all the species are, can be also somehow can be identified that Candida albicans has a different shape, parapsilosis are of the different shape. So by the view of microscopy, you can have some idea also. These are the chrome agar results of the quality control. ATCC cultures were used in the for these experiments. And these are basically the PCR to RFLP results of quality control ATCC culture. In this, you can see that Candida albicans shows the 2 bands of different sizes. And there are also most of the species given the 2 bands of different sizes. And some species are not cut and given the same size as we got from the PCR results. These are the results of gel electrophoresis of 17 yeast species we identified in the study. The prevalence of the Candida species, we got almost 18 Candida species identified in this study. 40% were Candida albicans. 32% were Candida tropicalis, followed by Parapsilosis cruzi, glabrata, and femita. 6% were other species that were the rare species, and 1 pathogen was identified in each case. 2 mixed yeast infections were also identified, which were the combinations of Candida albicans and glabrata. It is very important because it is very crucial that to identify the mixed species. If in a culture there are the mixed species, it is very crucial for the correct treatment. You can see here that in PCR, we got 2 bands So it identified that there are the two species, possibility of the two species here. And that was confirmed by the results of our FLP when we analyzed the results, that they show that they were the combination of glabrata and albicans. 3 rare species were also identified using Singer's whole genome sequencing, naming Cotemia ohemeri, Durkheimia, and Werker-Milia. Very difficult names. Actually, they are the Cotemia is first time reported in this study. As about the specimen types, most of the samples were collected from blood, and second most prevalent specimen was urine, from which the infections were collected, followed by pus, tissue, HVS, and tracheal aspirate and other fluids. As about the patient demographics, most of the samples were collected from the age 2 to 17 years, and followed by 18 to 29 years, and 60 to 69 years. And as about the gender distribution, most of the infections were identified in the males as compared to females. This slide is showing the correlation between fungal species and specimen type. You can see that Candida albicans, Femeta, and Cryptococcus mostly were identified in the blood. And so on, you can see Candida krusei and tropicalis were most common in the urine. A little bit discussion about the results, that a few points I want to highlight.
Your time is up. You have used all your 10 minutes. Can you just?
Just finishing. Most prevalent species was Candida albicans, as was reported in the literature, and the results are the same as as mentioned in other papers. But the trend of most common non-Candida albicans species is a little bit different in Pakistan as compared to the other regional studies. Candida tropicalis, we found the most prevalent non-Candida albican species, as compared to in the literature where the Candida glabrata was the most common non-albican Candida. The ratio of mixed yeast infection is also the same as mentioned in the study, other literature, and the ratio of specimen types and gender distribution is also same as mentioned in most literature from other populations. This is the first reported incidence of Cotemia in Pakistan, and patient data was unavailable. This study eventually is already validated previously in my PhD and many other studies by other authors from other areas and regions of the globe. Concluding my results, we have a few statements that this method can identify all medically important common yeast infections except the rare species, and it is very highly sensitive and specific, accurate, cost-effective DNA-based identification method. It requires minimal expertise, and clear band patterns facilitate easy interpretation. It enhances the Candida infection diagnosis and management, and this method proved to be the best solution for rapid, accurate molecular identification of yeast infections. Cost of the test is we have estimated very low. It is almost 1,000 rupees we have calculated. And home taking message is that this approach is ready to use in the routine clinical diagnosis.
That's enough, Dr.
And
you have used all 12 minutes now, 10 minutes.
in hospitals to benefit poor patients of lower middle income countries. Thank you. Thank you. If you have any questions, I am here to answer.
Thank you very much, Dr. Humera. It was an interesting work and interesting talk, and these kind of basic research is important to develop such diagnostic tests for the lower and middle income countries. Thank you.
Thank you. Thank you.
So thank you very much, everyone, for being patient with us and staying with us till the end. We've concluded the session. The results for the best presentation will be announced tomorrow during our final session. Thank you.
Thank you.